Hi everyone. In this video, we're going to look at the basic principle of cell signaling pathways and the types of receptors that can be involved. Regardless of the cell type, specific signal, or required response, all cell signaling pathways follow the same basic steps. First, a signaling molecule, which we call a ligand, binds to its receptor in a process termed reception, because the cell is receiving the signal. This typically causes a conformational change in the receptor, which activates a series of relay molecules in the process of transduction. This concludes with the activation of a cellular response, which could be a variety of activities, such as turning on a gene, transporting something, moving the cell, or even cell division. The specific response will depend on what the signal is and what type of cell received it. There are three main types of plasma membrane receptors, and the first one that we're going to look at here is a ligand-gated ion channel. The name here is pretty self-explanatory. It's a channel for an ion, which can be opened by a ligand. When the ligand binds to the receptor, the channel is opened, allowing the flow of ions down their concentration gradient. Channels of this type are found throughout eukaryotic organisms, such as those which act as receptors for neurotransmitters, allowing the passing of information from one neuron to the next across the synapse. Our second type of plasma membrane receptors are receptor tyrosine kinases, which frequently function as receptors for growth factors. In order for this type of receptor to function, there need to be two of them working together. Once each receptor has bound a ligand, the receptors will dimerize. If we zoom in to the intracellular portion of the receptor, we can find a whole host of the amino acid tyrosine, indicated by the letter Y here. The receptor will then autophosphorylate, or add phosphates onto itself, and only after this is it fully active and able to activate the downstream signaling pathway. A third type of plasma membrane receptors that we're going to look at are the G-protein coupled receptors, or GPCRs. These are the most diverse group in terms of function, but they all work by the same basic principle. As with the other types, it starts with a ligand binding to the receptor, which, as the name suggests, is coupled to a G-protein, so called because it binds to and hydrolyzes GTP. Once the receptor is activated by the ligand, the G-protein will exchange its bound GDP for GTP and will then activate an enzyme embedded in the membrane. The enzyme will begin catalyzing its reaction, forming another signaling molecule called a second messenger, which can diffuse through the cell to activate its own effector proteins. This will continue until the G-protein hydrolyzes the GTP to GDP, causing the G-protein to move back to the receptor, ready to start again with the next ligand. Since second messengers are so important in biology, let's have a look at a couple of specific examples. First, we're going to look at cyclic AMP. As with our previous overview, we start with our ligand, which binds to our receptor, causing activation of the G-protein. The enzyme activated in this case is called adenylyl cyclase, which converts ATP into cyclic AMP. One of the major effector proteins activated by cyclic AMP is protein kinase A, which can then go on to phosphorylate its own set of proteins, leading to the cellular response. The other second messenger we're going to look at is calcium ions. This is even more common as a second messenger than cyclic AMP. Again, we start with our ligand, which, as with the previous example, activates the receptor and subsequently the G-protein. The enzyme in this case is phospholipase C, which breaks down one of the phospholipids in the plasma membrane into diacylglycerol and inositol triphosphate, or IP3. The IP3 is itself a second messenger, which opens channels in the smooth ER membrane, releasing calcium ions into the cytosol. Now, since the normal cytosolic concentration of calcium is very low, this rapid release causes a significant change in the concentration, leading to a range of responses, including regulation of enzymes, cell motility, and muscle contraction. Now, so far, we've only considered receptors on the plasma membrane, and that's true of all of those with hydrophilic ligands. There are some receptors, however, which have hydrophobic ligands, like steroid hormones. Since these molecules are hydrophobic, they're able to diffuse straight across the plasma membrane and bind to receptors inside the cell. Once bound to their ligands, these receptors often function as transcription factors, moving into the nucleus, where they can bind to DNA and activate transcription of specific genes, leading to the production of new proteins from the instructions in the mRNA, which can then alter cellular functions, allowing the cell to respond to the signal it received. Thanks for watching, and please be sure to check out the video on signal amplification and regulation.